Please join me in welcoming to the Distinctive Voices podium, Dr. Monica Deuce.
Good evening, everyone. Thank you for the introduction, and thank you for being here today. I'm really honored to be giving this lecture. So today I'm going to tell you about the work our lab does. And I want to begin with the question I feel, for me, it's one of the most fascinating questions in biology, which is how the environment shapes our brain and our behavior. Now, in the past, scientists have talked about nature versus nurture, how the interaction, or really the fight between our genes and our environment shape who we are. But today we really talk about nature and nurture. And I don't know. Okay. We really talk about nature and nurture, how genes and environment work together to shape our behaviors. And in fact, we know that the way they work together is that the environment actually that the environment changes the way our genes, the sequence in our DNA, is expressed. So it works like a switch. And this field of study, it's called neuroepigenetics. When we talk about the environment, we think of very specific things. We think of adverse experiences that we experience during child development or adverse experience that we encounter as adults. But what is true is that the environment is really everything around us. The environment might be what material the water bottle you're drinking from is made of. It can be the temperature of the environment. Today it's beautiful. I come from Michigan. It was, I think, 15 degrees Fahrenheit there this morning. And it doesn't just have to be the interactions that we have with each other and that shape us. And in fact, one of the biggest environment that we really live through every day is what we eat, is our diets. And so today we're going to talk about our food environment, our diet influences our behaviors. And now, I don't have to tell you, you know from your own experiences that the food environment you're in really changes our eating patterns. So we know that when we are in environments where there are certain types of foods, especially those that have high quantity of sugar, salt, and fat. Now, my favorite food is pizza, and I am Italian. But we know that when we have foods that have a lot of salt, sugar, and fat, we tend to overeat. Now, this didn't really make sense to me because we know that evolution has shaped these very elegant systems where we can keep essentially track of how much energy we have in our bodies. When the energy is dwindling, the animals is hungry, goes to look for food, to forage, and eats, and so now we have more energy. And then when we've accumulated enough energy, we're seated and we stop eating. But I'm the mom of two bichon frisés. This is Cupcake, and the other one is Sprinkles. And it occurred to me how naive the idea of this scale was when my bichons ate 10% of their body weights in chicken jerky because I forgot a bag of chicken out. It's clear that in the presence of some foods, whether it's pizza for me or is chicken jerky for my bichons, this homeostatic mechanisms, the scale doesn't work as well. So, of course, why we overeat is a question that a lot of scientists are studying today, and I put the names of 3 of my favorite ones here. And one of the ideas is that some of these foods just affect our decision-making, so we might not be able to stop when we ought to. And other scientists think that actually they affect the way we perceive nutrients inside our bodies in terms of hormones, so we might not actually know how many calories we're eating. But there is a third area of investigation that has emerged more recently, and that is how the dietary environment is perhaps affecting our ability to perceive these foods. So we know that our senses, such as Taste, smell, and touch are really our windows onto the world. This is how we perceive the world, not just us, but all kinds of animals. And especially we know that taste and smell are really important to determine what you're going to have for breakfast, lunch, and dinner. And we know that it's really some of the innate qualities of foods that really determine how much we're going to eat of them. And the quality of foods that we tend to overeat are usually food that are sweet, salty, and fatty. And that is because we are born with innate preferences for some food qualities. And you don't have to think of your own experiences. You can just look at the expression of babies when they first eat their first broccoli or their first fruit. We are born to love some qualities of our food and to really dislike others. And so the way we perceive this quality is clearly very important. Now, since the perception of this quality using food is really important, some people thought that perhaps in people that overeat and gain weight and become obese, maybe they're enjoying the taste of food too much. But in really the last decade, the research has emerged saying exactly the opposite, and that people with obesity actually perceive some of the qualities of food less intensely than people who are lean. And now, even if the research is still undergoing and somewhat controversial, not all the studies are finding these changes in taste with obesity, it's clear that what we thought was true, that people who are obese just enjoy their food more, is not true. So the question I really want to talk tonight is whether diet alters our sensory perception of food and whether this has an effect on what we eat. In fact, we already have some of the answers to this question, and we know that diet composition changes the way we perceive the sensory qualities of food and affects our food intake. And if you were around about 30 or 40 years ago, you remember all this public health campaign of salt. We know that depending on how much sodium we have on our diets, that changes our perception of the salty qualities of food. The more sodium we have, the less salty we taste, and so we usually sprinkle more salt in order to feel the saltiness. So more salt decreases our perception of saltiness and increases our intake of salt. We also know that some of this is true for fat. So the amount of fat we have in our diet also decreases the perception of fat in our mouths. And then we actually don't know as much about sugar, whether the amount of sugar in our diet alters the ability for us to taste sugar. So I was really interested in this question because, of course, one could argue that the sugar is perhaps one of those three components that really changed the most in our diets, because we went from eating sugar in this form to eating sugar in this form and drinking it in this form in really the span of my lifetime. Now, the reason sugar is now everywhere is that it's not just anymore a naturally occurring substance, but it's being crystallized and added to our food as a food additive. So if we look at the foods in American supermarkets, about 75% of foods have added sugar. And this includes foods that we usually think of as healthy, like granola and yogurt. 60% of yogurts in the supermarket have added sugar. Now, you could say, okay, well, I can look at the label and figure out how much sugar there is in this food. But it turns out, if you look at the food label, sugar comes with many different names. I think right now we're up to 60. So sugar doesn't just come with the word sucrose or glucose or agave syrup. It comes with many different names, and it's often hard to identify it. Okay, say you now recognize all the 61 names, then how would you know that you're eating enough? It turns out that if you look at the food label, if you look at food labels, sugar is the only macronutrient for which there isn't a recommended daily value in food labels. So if you look at the food labels, and for instance, for fat here, we now know that this granola bar has 3 grams of saturated fat. That's 15% of the recommended daily value. But notice when we look at sugar, here, there's nothing next to it. So that's quite interesting. You know, you essentially cannot know by looking at food labels how much sugar you're having and how much you should be having. Now, there are set guidelines for sugar, and the guidelines say that really we should eat between 6 or 9, depending on whether you're a male or a female, tablespoon of sugar if you're an adult, but actually this is how much we in America tend to eat every day, so 17 to 21 tablespoons, and when it comes to teens, it's even more. Now, so we are overeating, we're eating more sugar than we're supposed to. And now this is not only because you might have cake for breakfast, right? It's actually because, like I've told you a minute ago, sugar is everywhere in our foods, in our supermarkets. And so yes, if you have a muffin for breakfast, you'll essentially eat your daily allowance of sugar. But you might not know that actually a Gatorade has more sugar than a muffin. You might not be thinking about that unless you're really proficient at reading food label. Things like smoothies and pasta sauce, and like I said before, yogurt and even bread contain added sugar. Now, because we're eating so much sugar, we eat about 130 pounds of sugar per year per American. This is an upper estimate. The lower estimate is somewhat around 70 pounds. And this is really kind of a new thing because this is how much we eat today, but in 1820, we ate for the amount of 1 can of soda in over 5 days, whereas now we essentially eat a pound of sugar, almost a pound of sugar every 5 days. Now, as Americans, we're not doing great. We are only supposed to have 5% of our calories from sugar, but we end up eating actually 20 to 30% daily calories. But as you can tell, the rest of the world isn't really doing much better. Now, sugar is bad because we know that, or having a diet that's high in sugar is bad because we know that it's highly correlated with weight gain. And so if we look at the map of the incidence or the prevalence of overweight in the United States, we essentially see that 8 in 10 people in a room will be overweight, and about half of those people will have obesity. And of course, that's bad because that increases the incidence for chronic diseases like diabetes and heart disease. So the question of whether dietary sugar leads to changes in taste and possibly change our feeding pattern is a quite important one, and one that my lab is investigating. Now, we're studying this question in fruit flies. So why fruit flies? Well, it turns out that the taste system in fruit flies is much, much simpler than those of humans or either rodents. So the taste system in fruit flies has this conserved neurochemistry and it's organized along similar principles, but it's much simpler. And it's because flies eat with their proboscis, and they have their taste buds on the outside of the proboscis. So here's a fly brain. You can tell, probably imagine this is where the eyes would be. And they have their antenna where they sample the environment on the, here on the proboscis, and then they send the signal directly to the brain. So it's a very simple and elegant system. And in comparison, this is a rat taste system is quite different. We have 3 different kinds of taste buds, 3 different nerves that bring the taste information into the brain, and a lot of other passages once the information is in the brain. So if we study fruit flies, we have a much, much simpler system. Also, you know, because fruit flies are in your kitchen eating your bananas, that they eat what we eat. They're omnivores like us. They're very inexpensive compared to other model organisms in the lab. They grow fast. We can do a lot of experiments with them. And fruit flies indeed have a brain. The brain is very tiny. It's about as big as a sesame seed. But even with the brain that tiny, they have amazingly complex behaviors. You might actually remember that last year, the Nobel Prize for Physiology and Medicine was given for the discovery of the gene and circuits that controls circadian rhythms and sleep, and it was in flies. And by studying flies, we've learned about the genes that control aggression. So fruit flies, males, in the presence of a female and scarce food, they will fight each other. So you'll see here boxing. Here's a female. She's right here doing science, and here two males fighting each other for her attention. Fruit flies have also been studied to understand and discover the genes and the neural circuits that underlie addiction. So this is, again, a male fruit fly that was given a little too much alcohol. And as you know, fruit flies will walk and fly really fast, but this fly has a lot of trouble climbing up its vial. This fly is highly inebriated. It cannot really control itself. And at some point, if we watch this whole video, it would actually pass out on the side of the vial. So fruit flies are used to study addiction, and even for more complex social behaviors, and even to understand the difference between the male and female brain. This is a strain of fly with a mutation in a gene called fruitless. When this gene is mutated, males no longer court females, but only court other males. And they've developed this new courtship behavior, this way of serenading each other, which involves this elaborate dance where essentially they chain one after the other. And you'll see flies sticking up their wings at some point, and that's actually, they're using the vibration of the wing to serenade the mate they want to attract. So fruit flies are extremely complex. They have complex behaviors, and yet they're simple enough for us to be able to understand the genes and circuits that determine those behaviors, and that's why we study them in the lab. And so that's also why we study them in my lab, to understand how dietary sugar leads to changes in taste sensation and whether this determines changes in feeding patterns. So how do you make fruit flies obese? How do you give fruit flies extra sugar? It's actually pretty simple. Fruit flies eat some sort of porridge. It has cornmeal and sugar and a little protein. And what you do, you just add extra sugar in their food. So we add 30% sucrose. And you might ask me, Monica, how much is 30% sucrose? Is that a lot of sugar? Well, the sweetness of 30% sugar is actually close to the sweetness of a chip Ahoy cookie, which I'm pretty sure almost everybody in this audience has had. And so it's a lot, but it's not too much, because if we look at the sweetness of many of our desserts and the majority of children's cereal, they actually have over 60% sugar. Right, and our control diet is 5% sucrose, which is the sweetness of strawberries, so not very sweet. When we give our animals a sugar diet for different days, CD stands for control diet, and we measure essentially how much fat they have, we see that the more days they eat this diet, the more essentially obese they become. They accumulate fat very much like we do. And then we can measure how much they're eating. So if we look at the flies that had the strawberry sweetness diet or the 5% diet, they pretty much eat the same amount every day. However, and so these are different days, and we're measuring the licks, how many licks the fly are doing in each of those two foods. But you'll see that when we give them the high sugar diet, they essentially increase their feeding behavior over time. And this is very similar to what happens to rodents and possibly what happened to us. So when we give flies high dietary sugar, they also overeat. So we give them a lot of sugar and they overeat and develop obesity. So the question I wanted to ask is whether they also develop a blunting or a dulling of their taste. So how do you measure fruit fly taste? I've told you that fruit flies have their taste buds in the proboscis, so we trap them inside a little cone, and then we touch their proboscis here with a sugary solution. If they extend the proboscis, they can taste it, like in the video. If they don't extend it, they can't. And so because we're scientists, we want to quantify this behavior. We can give it a score from 0 when there's no extension of the proboscis, the flies cannot taste, to 1, like in here, when there is a full extension. So we trap the fly in this little cone, and then we give them, we touch the proboscis with different concentration of sugar, something that's as sweet as zucchini, as sweet as strawberry, or as sweet as soda, and then measure their responses to the sweetness of these foods. And as you might imagine, as it makes sense, they respond more to things that are sweeter than to things that are less sweet. Now, when we give them a high sugar diet in dark red here, you see that there is a decrease in their responses to sucrose. Now, this is not a motor defect. If we touch their proboscis with other things, they can taste fine. And in fact, we know this is a specific defect in the taste of sugars. And then we can look what happens maybe a little earlier, 3 days, and we find that there's this intermediate decrease. So when we put flies on a high sugar diet, they become obese, they overeat, and also we observe a decrease in their ability to actually taste the sugar. So the first thing we thought, it's like, well, okay, maybe the taste buds that respond to sugar are just dying. So what we did, we actually label the sweet taste cells with a protein that is green. We just tag them with something that's fluorescent and green, and then we counted. You can see here's the cell body. This little foot here is actually the antenna of the cell. This is how they sense the environment. And then this little cable here is what would go into the fly brain that I've shown before. But when we compare the number of cells between the flies in a control diet and a high sugar diet, absolutely no difference. So good news, the cells are not dying. So if they're not dying, are they perhaps responding less potently to sugar? And is that why the flies cannot taste? So to test this hypothesis, we took a fly and we glue it to a slide, and then we open a window in its head, put it under a microscope, and stimulate its proboscis here with a sugar solution to look at the activity of those taste cells in this animal that's alive. So you might ask me, what are you doing again? You're cutting a piece of cuticle in the fly head? How does that look? This is how it looks. So red eyes, so these are essentially, you're looking at the fly from the top here. These are the eye. We've glued it here. Actually, we've waxed it. And then these are the antenna, which would be here. And this white part here is actually the brain. So it's a blob of white. And then how are we able to see the sweet taste cells? We're able to see them because we tag them again with a green fluorescent protein. But this is a different type of protein than the one I showed you before. It's one that becomes more green, goes from dark to green when the neurons are active. So we can use the fluorescence of this protein as a proxy to measure the activity of the neurons. And this is what it looks like. So here's a video. What you're seeing here is essentially the neurons flashing. The more gray is essentially this protein becoming more green, just as in showing a grayscale. And so here is, you're looking at the activity of the taste neurons in the fly brain responding to sugar stimulation of the proboscis in real time. So when we did this, and we measure the responses of the taste neurons to sugar in the flies that were given a controlled diet or a high sugar diet, we measure about a 30% decrease in the activity of the cells, meaning that the taste neurons, the sweet taste neurons of the animals that are on a sugar diet are less efficient. They don't respond as robustly, and this is why then the fly is not tasting the sugar. So far I've told you that when we give the flies a high sugar diet, we get a decrease in sweet taste sensation, and I've also told you that the flies overeat and become obese. But we wanted to know whether there's a connection between these two things. Are the changes in taste driving the overeating and the development of obesity? So remember that on a controlled diet, flies can taste fine. On a high sugar diet, their taste is decreased, they overeat. And we know that this occurs because the neurons don't respond as robustly to sugar. So what would happen to the feeding patterns of these flies if we now fix its neuron so that it doesn't experience a decrease in its sweet world? Will it, now that it can taste, continue overeating, or will it behave like a fly that was on the controlled diet? So to do this experiment, to activate the neurons, we use something called optogenetics. So optogenetics uses light to turn on neurons. And the way you do that is that you can use genetics to put a channel that is only activated by light in the cells of your interest. In our case, is in the sweet sensing cells. And then when you shine light, you get a depolarization, a change in the voltage of the membrane, which sends the electrical signal that will activate the neurons. And so we put this light channel only in the sweet sensing cells that we've been talking about, and we only turn on the light when the fly was pecking or eating at the food. When we did that, the animals that never got any light and that were on a high sugar diet overate on the high sugar diet food, but the ones where we shone the light to keep them from experiencing a change or a decrease in their taste, look at this, they don't overeat and they don't develop obesity. So this strongly suggests that the changes in taste are actually driving the changes in food intake, and that when we fix these changes in taste, I'm showing you how we fix them with light, but we have other ways to fix them, we actually get flies that, even if they're eating the high sugar diet, can still taste, and they have normal eating and remain lean. So changes in sweet taste promote overconsumption. But remember at the beginning, I've told you that when we give the animals a high sugar diet, even after a few days, they already gain a little weight and accumulated fat. So how do we know that the changes in taste are really a consequence of dietary sugar rather than a metabolic consequence of obesity, right? You might imagine when people develop obesity, animals develop obesity, there are changes in their physiology of the body, and we know they can develop, for instance, diabetes because their pancreas doesn't work as well. Maybe something similar is happening with the taste cells. Maybe it has nothing to do with sugar itself, but it has something to do with the fact that by eating sugar, the flies are gaining fat. So to do this experiment, we essentially try to uncouple the diet from the obese state. And the way we did that is by using fly genetics and by using flies that are genetically obese, even without exposing them to diet. So remember, these are our control flies. On a controlled diet, they can taste fine. On a high sugar diet, they develop obesity and they have a blunted sense of sweet taste. Now we use flies that are genetically obese. So these flies, even if they are eating just their normal control 5% sucrose diet, they're already as obese as the control diet, as the control flies on a high sugar diet. And then when we give them a high sugar diet, they become even more obese. So if we thought that it was just the obese state that was driving the changes in taste, we would expect these flies, which remember have the same amount of body fat as the control flies that have been eating sugar along, we would expect them to be unable to taste as well. But what we find instead is that animals that are genetically obese, they're totally fine. They can taste normally. And it's only when we give them a high sugar diet then they experience a change in their taste function, suggesting that obesity alone, in the absence of the cupcake, in the absence of the high sugar diet, has no effect on sweet taste. So then we can do the converse experiment, and we can use a fly that's genetically resistant to obesity. So these flies, no matter what they eat, whether they have the controlled 5% diet or the high sugar diet, which is the 30%, they remain lean. They always look like the control flies on their control healthy diet. Now, if we thought that obesity was driving the changes in taste, we would expect that these flies, because they remain lean, even if they're eating the high sugar diet, they should be able to taste. But in fact, what we found is that these flies, even if they don't become obese, even if they stay lean, they are still unable to taste. And so in all our experiments, we only ever saw changes in taste when we gave the flies a high sugar diet independently of obesity, suggesting that it's really the dietary sugar that's driving the changes in taste rather than these changes in taste being a metabolic consequence of the obese state. Now, of course, when we talk about sugar, there's really two components of it, right? Sugar, it's sweet, and sugar has calorie. So is it high dietary sweetness? Is it the fact that the diet is so sweet that it's turning down our sweet sensing cells? Or does it have something to do with the actual sugar that's present in the extra sugar that's present in the food? Well, to do this experiment, we gave flies essentially a high sweetener diet. So we gave them the normal food, but gave them extra Splenda in their food. So it tastes sweet, but it has no extra calorie. And when we did that, so in yellow here, and we measured their responses, their taste responses to now sucrose, notice that there's absolutely no difference. So it's not just the dietary sweetness, but it's actually the levels of dietary sugar in the diet that are altering taste function. So today I've told you that when we give flies a high sugar diet, we observed a decrease in taste and an increase in eating and the development of obesity. And when we essentially take flies that are on a high sugar diet and we put back in genetic ways the proper taste function, so animals never experience a decrease in their sensory sweet world, we get animals that are lean and do not overeat. And I've shown you that it's really dietary sugar rather than dietary sweetness, and that this occurs independently of obesity. So when we go back to the slide that I showed you at the beginning, when we talked about salt, fat. Now we can also add listing flies, sugar, and talk about how the composition of the diet is really important in determining what we eat, because it can change the way we perceive those foods. And of course, all the work that I've shown you today was done in flies, and it raises the question of what happens in us. But I've already told you that there is evidence out there that humans that are obese are experiencing changes in taste, a dull and a sweet taste, and that actually when we put people on a diet that has less calorie, specifically less sugar calorie, we see an increase in their perception of sweetness. So while we don't know what happens in humans, there is, it's an interesting question, and there is some data already suggesting that perhaps this could happen in us too. So for the last 5 minutes, I want to talk about 2 questions that we're following up that arise from this work. The first question is, how do changes in sweet taste promote overconsumption? What is the link between changes in sweet taste? Why would they make flies eat more? Well, we know actually that humans that are obese get less reward for the food they eat. And so there are a lot of studies of this. Some of the most famous are those, they're called milkshake studies, where essentially humans are given a milkshake every day for 2 weeks. And then scientists look at the responses of the brain to milkshake. And if you never had milkshake, then your brain is on fire. It's like, oh my God, that ice cream and milkshake. But if you've had a milkshake every day for 2 weeks, there's very little activation. So again, this is counterintuitive, right? We usually think that people that are overweight just enjoy their food too much, but exactly the opposite is what scientists find. So is it possible that the food feels less good because it doesn't taste as intensely as it used to? So is there a connection, right, between the changes in taste and the changes in reward or pleasure that we get from food? So again, we can go back to the fly and try to ask this question. We can have a fly here with a normal diet, always eating bananas. One day, it eats a cupcake and it's amazing. It has all this pleasure. On the other hand, we have our high sugar diet, our fly eating the high sugar diet. We know it has a decrease in taste, and so the cupcake doesn't taste as sweet and doesn't feel as good. And is it possible that the difference between what the cupcake feels like now and what the cupcake used to feel like before, so this is the gap that we fill, or that the fruit fly and we fills by overeating more food to get to the same level of pleasure that it used to get before? And so this is a question that we are asking in the lab now. The other question, of course, is how does dietary sugar change the physiology of the taste cells? I've told you it's not sweetness, is the amount of sugar that the body is seeing. And I find this to be really fascinating. So we know that nutrients and macronutrients are really important for our body. We know that they're really, really important for the physiology of the cells because they're essential. We eat, nutrients go inside the cells, a bunch of things happen, and then the energy is used up, and then there is waste that comes out. But it turns out that metabolites and nutrients have a much more direct impact on the physiology of the cells than just being used for energy. They're actually a source of information, and the information they're giving to the cell is the nutrient environment. And so if we look at the basic central metabolism, which you might remember from school, where glucose is broken down into 3 carbons through glycolysis and the TCA cycle, all the metabolites that are made in the central pathway in every cell, these are different names for them, these are actually used by complexes that modify our DNA to turn on and off different genes. So the activity of the molecular machines that determine when, how much, and where a gene is expressed in your body are intimately linked with the nutrient environment that the cell finds itself in. So an example of this is when you're eating food, my second favorite, pasta here. There's a lot of folic acid. Folic acid is then metabolized by the cellular machinery into different metabolites, in particular this metabolite here called methionine. Methionine is then used to essentially tag the DNA, shown here, with different tags that turn down the expression of the genetic code that's found around these tags. So then the more pasta or the more things that will be metabolized as methionine you get, then the more of these little lights, street lights you get in the DNA, to stop or halt or decrease the expression of the genes. Now, these effects of metabolism and gene expression don't just last for an hour or a day, they can actually last for a lifetime. And this is why the nutrient environments we develop as kids is often really important to determine the incidence for chronic diseases such as diabetes, cancer, heart disease, and even neurodegeneration that we develop as adults. But not only it spans one generation, we now know that it can span multiple generations, and so that the environmental influences on our cells can really last more than our own lifetime. The most famous example of this is the Dutch famine study. The Dutch famine, also known as the Hunger Winter, occurred at the end of World War II when in the Netherlands, in Netherlands occupied by Germany, there was a blockade, and the Western territory of Netherlands didn't get enough food, essentially. And so during that time here, the amount in these critical months between 1944 and 45, the number of calories per day that people ate dipped to 1,000. So, of course, during that time, people kept on living, and some women were pregnant, and some of them conceived, some of them had babies in different developing stages during the famine. Some of them were in the first trimester when they occurred, the famine occurred, some of them in the second, some of them in the third. Now, the children of the grandchildren of the mother, so the children of the children of the baby that were developing, and their grandchildren actually have this environmental scars from when their grandparents experienced a famine. And depending on when they experience a famine, they develop an increased incidence for different types of diseases. If it was in the first trimester, they had an increased incidence of obesity, cardiovascular disease, hypertension. In the second semester, they had different types of diseases like renal disease and pulmonary disease. And in the third semester, if they experienced the famine as they were in their especially last few months, they had a high incidence of things like schizophrenia, depression, diabetes, and personality disorder. And there is a very nice lay science article in the New Yorker if you want to read about this. So this is perhaps one of the most famous examples where this changes in the nutrient environments can really impact many generations. Another interesting example of this that you might have encountered before is the agouti mice. So these two mice are genetically identical, but they don't look identical. The only difference here is that they came from mothers that were genetically identical, but both mothers were given food that had bisphenol A or BPA, the bad stuff in your water bottle, which is a neuroendocrine disruptor. But the mother of this mouse just got the normal diet for a mouse plus the BPA. The mother that gave birth to this mouse got the same diet, but extra B vitamins, B12, betaine, folic acid, and choline, which actually get metabolized into those little tags that I showed you before. So that if we zoomed in into the DNA of the yellow mouse, it's missing all these tags. And then if we zoom in the brown mouse, it has now all these tags that function as switches to change the expression of the DNA. And now the brown mouse turns out to be healthy, whereas the yellow mouse is prone to cancer and developing diabetes and becomes obese. So this idea that the nutrient environment is a form of environment and has a really big impact on our cells, but not only our cells, on our brain cells too, and might change what we eat, which further impacts our food environment. It's a new field of study that's called nutrigenetics. And it's a fairly new field, but really stresses the importance of diet and diet composition in our daily lives. And so we as scientists are studying this and asking all this interesting question, but then when we think of sugar, it's really kind of out of the realm of science and into our realms of public health. And the question for us is, what do we do about sugar? Is sugar the public health crisis of our time? We had a public health crisis half a century ago, and it was cigarettes. And so what do we do as a society? What is the right way to deal with sugar? And so this is a question I cannot answer, but is a question I think that a lot of people, and I'm sure that many of you in the audience, are also thinking about today. So before I give my things and take some questions, I want to tell you about a podcast that I do at the University of Michigan with a science writer, Liz Wazen. So it's called How to Science, and there are 2 seasons. And it's really about scientists rather than science and research per se. So it's about really trying to find out who are scientists, what are our lives like, and what makes us tick. And if you're interested in neuroepigenetics, so and you can find this wherever you get your podcasts, Apple Podcasts, Stitcher, Google Play. You can just put this in your search engine and you will find it. If you're instead interested in this idea between genes and environment and behavior, I actually teach a class about it at Michigan. And the final project of my students is actually to write an essay and make about a 10 or 15-minute podcast for the public about this topic. So if you're interested in these topics, you can just put neuroepic in the search engine, and there are about 30 podcasts that are made for the public about all these topics, including diet. So with that, I want to thank you for your attention. I want to thank the funding, both the philanthropies and the NIH funds that supported this work. A lot of this work was done by Christina May, who was my first graduate student in the lab. Christina is now trying to understand how taste changes the pleasure we get from food. Some of this work was also done by Anumit Vaziri, who's trying to understand how sugar changes the taste cells. Thibaut Pardo is studying how sugar is changing the way we learn and making memories about food. And Jasmine and Daniel are studying other different aspects of how sugar changes the regulation, the turning on and off of genes in our brain and how that affects our behavior. So thank you very much for your attention, and I'll take any questions.